students and welcome in the online session of class 9th Science. Chapter 2 is Matter Around Us Pure. Students, we have started the topic separation techniques. In that, three of the topics we have covered in the previous session. Now today we will see the separation techniques which are as follows: sublimation, chromatography, distillation, fractional distillation, crystallization and purification of drinking water. So let's start with the separation technique called sublimation. Now, sublimation is the process in which a solid directly changes to gaseous state. So, this is the definition for sublimation. So, in this process, any solid directly changes to gaseous state without coming to the liquid form. Generally, what is the routine that a solid first converts to liquid and then the liquid turn into gaseous state. But here in sublimation process, the solid directly changes to the gaseous state. So, it is important to know that some of the substances are sublimable substances. Why we are calling them as sublimable substances? Because they, on heating, they directly change to gaseous state. And the examples are ammonium chloride, camphor. Camphor is the kapoor which we used in our houses. Then naphthalene, iodine, anthracene. These are some of the substances which are sublimable in nature. That means when they are heated or when they are kept open in space, open in the space, they directly change into gaseous form. Like naphthalene balls you might have seen in your homes. These naphthalene balls are kept in the clothes also for fragrance. So after some time you will see that those solid balls decrease in size. Why they are decreasing in size? Because they directly change to gaseous form and so the size of the ball decreases. Now, the process of sublimation can be used to separate the sublimable volatile components from the non volatile components of a mixture. Now, suppose if you have a mixture in which some sublimable substances are present and other substances present are non sublimable substances. So, the method which can you use to separate the components is sublimation. Suppose you have the mixture of common salt and ammonium chloride. Ammonium chloride is also a white color powder which looks like common salt only. So what you have to do is, now this mixture you have to keep for heating. So here in the diagram you can see that there is a china dish in which we have taken the mixture of sand and ammonium chloride. You can take here common salt also, common salt and ammonium chloride. But sublimation technique can only be used when one of the component should be sublimable in nature. Otherwise, there is no use of doing sublimation technique. So we have to directly heat the mixture now. So while heating, what will happen, children? This ammonium chloride is sublimable. So on heating, this solid will directly change into gaseous form. Now in this experiment, what has been done is that a inverted funnel is kept on the dish. Now you can see this is the funnel. Now it is kept inverted, fine. And on the upper side of the funnel, a cotton plug is fit so that the vapors cannot go outside, fine. So you can see here that at this region, the ammonium chloride on heating when changes to vapors, now these vapors get collected on the walls of the funnel, and you can see here blue color salt which is deposited. So when these vapors strike the cool surface of the funnel, they again change into solid form. So from here these ammonium chloride get collected on the walls of the funnel and the sand will remain in the dish only. So this is the process of sublimation in which the sublimable volatile means which can change into vapor. So sublimable volatile components can be separated from the non sublimable substances. So when you heat sand and ammonium chloride will turn into vapors, and these vapors when strike the cool surface of the funnel, they get deposited here in the solid form again. Okay, so this is the process of sublimation. Now let's move on to the next technique which is known as chromatography. Chromatography. Chroma means color. Chroma means color. Okay. So here in this technique, what we do, we separate the colored components of a mixture. Okay, one can separate the colored components of a mixture. If in a mixture you have some colored dyes or some other colored substances, so they can be separated by using chromatography technique. Now there are various types of chromatography techniques: thin layer chromatography, gas liquid chromatography, but these are higher versions of chromatography. The very simplest form of chromatography is the paper chromatography. So let us see the theory of this method. So when two or more different substances may be soluble in the same solvent, but their solubilities are different. Okay, suppose in one solvent, here you have taken a solvent in a glass, it could be water or any other solvent. Now, two different substances you have mixed in this solvent, suppose, right? So a mixture is ready. So now these two substances you have mixed, but their solubilities will be different. One may be more soluble than the other, so their solubilities are different. Thus, when a solvent is allowed to pass over a mixture of such substances, the substance which is more soluble in the solvent moves faster and thus gets separated from the other substances. Okay, so this is the theory of chromatography. So here in chromatography technique, students, what is done? A mixture is taken on the chromatography paper, and when the water is allowed to move through that chromatographic paper, then what happens, the component which is more soluble in water will move along with the water and goes up to great distance. In this way, the components of the mixture can be separated. So the main principle to be keep in mind is that different substances have different solubilities in the solvents, and depending upon their solubilities they move along with the solvents up to different heights. So now let us try to find out this chromatography technique in detail. So what is the procedure, how this chromatography technique is done? So what we have to do, we have to take a thin strip of chromatographic filter paper. I'll show you along with the diagram also. Okay, so this is the thin paper chromatographic paper which we have taken. Now on this chromatographic paper we have to draw a line on the width side. Okay, along the breadth we have to draw a line. As you can see here, this is the line, and this line should be 3 centimeter from the bottom. Okay, now on this line we have to give a spot of the mixture. We have mixed the mixture in a solvent, and then we gave a drop, we just put a drop of that mixture on the line. Okay, and allow it to dry. Okay, so this is the first step. So take a thin strip of chromatographic filter paper, draw a line across the width about 3 centimeter from the bottom, put a small drop of ink. Okay, so now suppose we are we are separating the components of the ink, because in the ink different colored components are present. So we have to give the drop of ink at the center of this line and let it be dry. Okay, now when it gets dry, what we have to do, we have to take, now this is our one of the container in which we have taken water as a solvent. Okay, so this blue color which you can see downside is the water. So now this filter paper, this chromatographic filter paper, we have to keep in the container such that this ink drop remains just above the water. Okay, this drop of ink should not dip in the water, it should be just above the water, as you can see in the diagram like this. And then you have to cover this container. Okay, so this has become a chamber now. Okay, so paper inserted into the solvent saturated chamber. In a solvent chamber we have put the filter paper. Okay, so this is the clip by which we have hanged the paper like this. Now what will happen, children? Now it is a filter paper, so it has pores, it has small pores present on it. Okay, so now water will start rising up the filter paper. Okay, due to the capillary action. Fine, as you keep the filter paper on the water, filter paper starts absorbing the water slowly with the help of capillary action. Water will move through the pores and will rise up and will keep the complete filter paper wet. So now here water will start rising up in this way. Fine, and the components which are present in the ink, okay, they will also start moving along with the water, because these components are soluble in water, and according to their solubility in water they will move up to great heights. Fine, now you can see here that you get a drop here, then another drop you get here, then third spotting you are getting here, then fourth spotting you are getting here, right? So these four different components were mixed in the mixture or were present in the ink, which moved along with the water. And why this spot is up to this height and this spot is at a great height? Why? Because this component has moved up to this height. That means this is more soluble in water. This has reached only up to this low height because it is less soluble in water. So the more soluble component will move along with the solvent up to a great height, and the less soluble one will move up to lower heights. So in this way the components can be get separated. Fine. So now after developing after the different spots are got on the filter paper, we have to remove the filter paper from the chamber and let it dry. So after drying you can see the different drops like this. These are the different spots of the different components of the mixture. Okay, so these are the different spots of the different components of a mixture, and then with the standard colorization we can find out which is this component. Okay, so this is the paper chromatographic technique. Okay, so suspend the paper in a glass jar containing water so that the spot of ink on paper is just above the water level. As water rises up the paper by capillary action, it takes along with it the dyes particle. Okay, the dyes, the components which are present in the ink, they will also move along with the water. Ink is a mixture of two or more dyes. The colored component, which is more soluble in water, rises faster and produces a colored spot on the paper at a higher position. Okay, the one which is more soluble will move at more height and give a spot at the highest position, and the less soluble component rises slower and produce colored spots at the lower heights. Okay, as you can see in the diagram. So this is the complete step of chromatography, and at last the strip which we get on which different spots are shown, it is known as a chromatogram. Okay, it is known as a chromatogram. So this is one of the technique in which the colored components of a mixture can be separated. Okay, we have used paper chromatographic technique here. Now let's see the applications of chromatography, where chromatography is used. So it is used to separate the colored substances which are present in dyes and natural pigments. Okay, chlorophyll, if we have to separate the components of a chlorophyll, we have to use chromatographic technique. It is also used to separate and identify the amino acids. Okay, amino acids are the units of proteins which are present. So to separate and to identify the amino acids, again chromatographic technique can be used. Then to identify and to detect the drugs present in the blood. So this technique, you can see now how much important it is, as it is used in the forensic science to find out the criminals. Okay, so whatever drugs are present in the blood can also be found with the help of chromatography. Then to separate the different products of a chemical reaction, again this technique can be used. Now you can see here the spotted filter paper which is seen here is known as chromatogram. Okay, so like this the different components move uh, at different heights with the help of the solvent, as they have different solubilities in the solvent. So remember, all the components have different solubilities in the solvent, and when they mix with water they move at different heights along or on the basis of their solubilities. So this is about the chromatographic technique. Now the next technique is distillation. Now this distillation technique is used to separate two liquids. Okay, and very important thing is that in distillation process the liquid converts to vapors by heating, and then again the vapors are condensed by cooling method. Okay, this is the simple meaning of distillation. So distillation involves conversion of a liquid into vapor by heating, and then again by condensation method the vapors are changed into liquid form. So now when we have to use this distillation technique, so this distillation is used for the separation of components of a mixture containing two miscible liquids. Now if you remember, in separating funnel techniques we were taking two immiscible liquids. Now here we have to take a mixture of two miscible liquids. Miscible means which mix with each other. So such type of mixtures in which two or more miscible liquids are mixed, so they can be separated using distillation technique. But remember children that this technique will only work if the difference in the boiling point of those liquids will be between 30 to 50 degrees Celsius or 30 to 50 Kelvin. So this much difference should be there in between their boiling points. Otherwise, distillation technique cannot be worked out. Okay. So when we have to use the technique, distillation technique can be used to separate two miscible liquids only when they have a difference of 30 to 50 degree Celsius in their boiling points. If suppose one liquid has the boiling point of 100 degree, then other should be of 50 degree or 40 degree or 60 degree. Okay, not 90 degree. 90 degree Celsius if is the boiling point and other liquid has 100 degree Celsius suppose. So you cannot separate the two liquids by using this technique. So the difference between the boiling point of the two liquids should be 30 to 50 degrees Celsius. Fine. Now, what is the principle behind that? The separation of the liquid mixture into individual components can be achieved at their respective boiling points. Okay, so we have to heat the mixture. Whenever the boiling point of the liquid comes, it start changing into vapors. Okay, so the one which is more volatile component, more volatile means the liquid which has low boiling point will start converting into vapors first. Okay, and the less volatile will change into vapors afterwards. Okay, so the more volatile or the liquid which has low boiling point will start converting into vapors first, and so it can be collected firstly, right? And the liquid which has high boiling point will convert into vapors later, and then it can be collected. Now, suppose we have a mixture of water and acetone. Fine. So this is water and this is acetone. You can see here. Both looks transparent like this. Means if the label is not there, you cannot identify which one is water and which one is acetone except smelling them. Now, boiling point of water is 100 degrees Celsius and boiling point of acetone is 56 degrees Celsius. Okay. So you can see a good difference of boiling points here. Fine. So now how distillation technique is done. So here you can see a round bottom flask is there. Okay, so its bottom is round, so it is called round bottom flask, RB flask. Okay. Now, the mixture of acetone and water is taken in this round bottom flask. And you can see here a thermometer. Why this thermometer is kept? To see the temperature, whether their boiling point temperature is reached or not. Okay, if you know that at 56 degree centigrade, the acetone will start boiling and converting into vapors. So you will be ready with your setup so that now when acetone vapors will come, they can be collected here. Fine. So thermometer is a must here. Then with this round bottom flask, a condenser is attached. This one is known as a water condenser. Okay, so it has two outlets. Here you can see the water outlet is written and here the water inlet. Means from this side the cold water will go inside, and from here now this is connected with the tubes, okay, with the pipes to the tap. So water from the tap will come inside and will flow in the complete chamber, and from here the water will go out. Okay, why we are doing it so? Because the vapors which are coming from here, they are hot. Okay, so to cool them we need a water condenser here, and so running water is needed here, otherwise water will also become hot and then the technique will not work. So water should be floating here. Okay, will be rotating, regulating this side. So that's why we have an inlet pipe here and an outlet pipe that side. Now this is the water condenser, and you can see a pipe in between the water condenser. So vapors from here will come through this pipe and will pass through this middle pipe, and then when the vapors come here they'll get condensed with the help of this cool water here. So they will again turn into liquid, and then the liquid can be collected in the beaker. Okay, so now suppose you started heating it. So once you start heating, now we know that acetone boiling point is 56. Okay, so and water's boiling point is 100 degree. So which one of them will start boiling first? So acetone will start boiling first and will start converting into vapors. So as soon as 56 degrees Celsius temperature will come, what will happen? Acetone will start converting into vapors. Okay, water is still taking the heat, water is not boiled till now. So acetone will start converting into vapors. Now these vapors will move on through this pipe, and once they reach the condenser, there is the cool water here. So that cool water, when the vapors strike the cool surface, they will condense and again they will turn into liquid form. And then that liquid acetone can be collected in the beaker here. Okay. So till the complete acetone gets collected in the beaker, the temperature will remain here 56 only, will not rise up, right? Because due to the latent heat. Okay, so till all the acetone is not collected here, temperature here will not rise. Once all acetone comes out, the temperature again starts rising. And again, then when 100 degrees Celsius temperature will reach, water vapors will start coming out, which can be further collected in another beaker. Okay, so this is the method of simple distillation. Fine. So remember that the liquids should be miscible, and another important thing is that there should be a difference of 30 to 50 degrees Celsius in their boiling points, otherwise simple distillation will not work. Now, the next technique is fractional distillation. Now, this is the modified version of simple distillation only. Okay, the rest of the complete setup is same except one fractionating column is added here. So here also the two miscible liquids can be separated, but here what is the advantage here, that if the boiling point of the two miscible liquids of the mixture are very close, like if they have very close boiling points, we cannot separate them by simple distillation, but we can separate them using fractional distillation method. Okay, so the boiling points, if they are very close, less than 25 degrees Celsius or less than 25 K Kelvin, then we cannot use, a simple distillation. So in that case we have to use fractional distillation. Fine. So if the two liquids have very close boiling points, we can go for fractional distillation. Why this is so? That if the boiling points are very close, suppose you have taken in a mixture two such liquids, one is having suppose 30-degree boiling point and other one is having suppose 20-degree. Fine. So what will happen? As soon as 20-degree Celsius temperature will reach, the first liquid will start converting into vapors. Fine. But now 30 degrees also nearby, so the second also gets ready, and some of the molecules of 30-degree liquid will also start rising up like this. So both get mixed. Fine. That's why simple distillation cannot be used. So for that we have to use fractional distillation. Fine. Now, fractional distillation is carried out using fractionating column. Fractionating column has a long glass tube packed with glass beads or small stones. Let me show you the fractionating column. Now, here you can see the same setup, but you have a one different setup here. Okay, one more thing is added in the setup, that is the fractionating column. So this is the fractionating column. This is a glass tube only in which some glass beads are kept. Some glass beads are kept. So this becomes a fractionating column. Now what these glass beads are doing, children, these glass beads increase the surface area. Okay, so now suppose the mixture which is having a low boiling point will start converting into vapors. Along with this, the another liquid also starts changing into vapors. But the other vapors which whose temperature has not been yet received, so they will also, if they are also going, they will strike here with the glass beads and they will condense and again come back here. Fine. So this is the advantage of the fractionating column. Let me first complete this, then again I'll show you. Fractionating column has a long glass tube packed with the glass beads or small stones. The purpose of fractionating column is to increase the cooling surface area. So first thing, that fractionating column increase the surface area, and it also provide hurdles to the ascending vapors and descending liquids. Okay, so it provides obstacles, hurdles to the ascending vapors, the vapors which are going upside. So it provide hurdles. Okay, so unnecessary vapors which were rising, they will strike the fractionating column and will again fall down. Only those vapors will pass through the fractionating column who are required at that particular temperature. Okay, now let us discuss with the diagram now. Suppose you have a mixture of acetone and water again. Fine. You can take any other. You can take the mixture of chloroform and benzene. Any two liquids can be mixed and can be separated using fractional distillation. Fine. Now suppose this is the method in which acetone and water we are taken. Now what happens? Acetone has 56 degree Celsius, right? So it will start converting into vapors, right? So now acetone is going right this way. Okay, but suppose that some of the water molecules also get excited and they also start converting to vapors at 56 degrees, suppose. So this is the vapor of water which is going up, right? So acetone vapors along with water vapors, they are going in the fractionating column now. But the temperature is 56. So water should not go out. Otherwise, what will happen here? Water and acetone again both will mix in the beaker. So we have to stop the water vapors here only. So what will happen, children? These vapors of the acetone, because their temperature has reached, so they will directly cross the fractionating column. But the vapors of the water, because their temperature is not raised, so what will happen? They will strike with the fractionating column, and this fractionating column beads, they are little cool. Okay, so once the vapors of the water they strike these glass beads, they will condense here and again come back in the liquid form, and they will fall down again in the beaker. So in this way, there is no chance of mixing of acetone and water in the beaker here. So very pure acetone and water can be separated using fractional distillation method. Okay, so this fractionating column has a huge advantage because it increases the surface area. The rising vapors will strike the fractionating column and will fall down. Only those vapors can pass the fractionating column whose temperature has been achieved, and the pure substance can be collected on the other side. Fine. So water condenser is there, the same setup, only one fractionating column has been added here in fractional distillation. Okay, so the questions may come, what is the use of fractionating column? Fine, this can be the question because fractional distillation is much more advanced than simple distillation, and it is always better to use a fractional distillation than to go for simple distillation. Okay, now, let me show some of the fractionating columns which are nowadays used. Okay, so the simplest one, that we take a glass tube in which the beads were kept, glass beads. But nowadays there are different shapes of fractionating columns available in the market. Okay, so the fractionating column can be of this shape. So here also you can see this round shapes, they are increasing the surface area actually. So the vapors when go here, they strike the surface and they again fall down if the temperature is not reached. So it could be in this shape and structure, it could be in this, or this is the again glass bead type structure. Okay, so these are the different shapes of the fractionating columns which are used nowadays. Fine. Now, fractional distillation of air. Okay, so now we all of us know that air is also a mixture, and if one of us wants to separate the components of air, it is possible. Okay, so air is a homogeneous mixture of gases. Now, which gases are present in air? Nitrogen, oxygen, CO2, argon, etc. Now, these gases can be separated by using fractional distillation method. So, for this, the air is first compressed by increasing the pressure and then cooled by decreasing the temperature. Okay, so let me move on to the diagram. So, here you can see that air in means this is the filter from where the air is entering this complete setup, this complete machinery. So air will come on from this side. Okay, so again there, this is a chamber now in which the air is pressurized. Fine. So when air is filled in this chamber, this is the pressure, from these two rods the pressure is applied on the air in this closed chamber. So air is under pressure now, air get pressurized, due to which it becomes hot air. Okay, because temperature will then raise, if pressure is increasing it will become hot. Okay, because molecules they will be compressed, they will, their kinetic energy will increase, and so the air will become hot. Now this hot air, after getting pressurized, uh, this hot air will move through this tube and will come in this chamber. Now in this chamber, now freezing cold water is in. Okay, in this chamber you can see here this blue colored pipeline. In this freezed water is filled. So once the hot air comes in, this air also get cooled now. Fine. So this cold compressed air, okay, compressed air means it was pressurized, so it is compressed, it is in compressed form. So once the air is compressed, it will come in liquid form, it will come in liquid form. Because after applying pressure, if pressure is applied on the gases, they turn into liquid form. So now here cold compressed air is coming. Now this cold compressed air is allowed to go in this separator chamber. Now this separator chamber has the fractionating column. Okay, so First of all, the carbon dioxide comes out in the form of dry ice. The first component of the air which gets separates is the CO2, and it comes out in the form of dry ice. Okay, so this is out from here only. After this, when this liquid air is passed on to the another chamber, another fractional distillation column chamber, then the different gases start separating at different boiling points. Okay, now it is in liquid form. Once it reaches the fractional distillation column, now what will happen as the temperature is raised, so according to their boiling points they will start separating out. So the first gas which separates in this chamber is the nitrogen. As nitrogen has the lowest boiling point, so it will separate out first. Okay, and will be collected in another chamber. Then after nitrogen, argon is separated out. Okay, so let me check the boiling points. Nitrogen with boiling point minus 196 degrees celsius, then argon minus 186 degrees celsius, and at last oxygen has the highest boiling point minus 183. Okay, so you might be seeing that it is minus 196 this number, but remember the sign, it is minus 196. So this is the lowest temperature, and the highest boiling temperature is of oxygen. So oxygen will be separated at last. So at last you will receive the liquid oxygen here. Rest nitrogen, argon, they all will be separated out at their boiling points. Okay, so let me again revise you once, that once the air is let in a chamber where air is pressurized, so it changes into liquid form. So this hot air will move this side in a water freezed chamber, so it becomes now cold compressed air. So this is then passed in separator chamber where carbon dioxide is separated as dry ice. Now when this mixture is now passed on to the fractional distillation column, where the different gases are separated according to their different boiling points. Okay, the gas which has achieved its temperature will be separated out. So first nitrogen is separated out, then argon, and at last oxygen, which has the highest boiling point, is then converted into liquid oxygen. Okay, so fractional distillation method is used to separate the components of air also. Fine. Now, the next one is crystallization. Okay, crystals. Crystals means the solids which have a definite geometrical shapes. They are called the crystals. Like you might have seen the cubes of the sugar. So they have a particular geometrical shape. So they are called the crystals. Fine. So crystals are the purest form of a substance. In the crystal, there is no impurity mixed. Crystals are the purest form of the substance, and they have a particular definite shape also. Okay, so the process by which an impure compound is converted into crystal form is known as crystallization. Okay, so what is crystallization method? It is the process in which any impure compound is converted into pure crystal form. Okay, and the process is called crystallization. It's a very simple process. What we have to do, we have to take the impure salt, impure compound, whatever it is. Suppose here I am taking copper sulfate. So this copper sulfate is in impure form. Many impurities are mixed in it, suppose. So in the first step, what we have to do, that dissolve the mixture in water. Okay, dissolve that compound in water. Now, after dissolving it, then filter it. Okay, so after filtering, what will happen? The insoluble impurities will be separated out. Okay, and after filtration, what you will get? You will get a filtrate like this. Okay, so now what we have to do, two times filtration can be done. Okay, so first time what we have done, first time we have mixed it in water and then we filtered it using a funnel and a filter paper. Here is the filter paper, so you'll filter it and you will get downside, the liquid which you will get is known as the filtrate. Now you are heating this filtrate. Okay, in the next step you have to heat the filtrate. After heating, what will happen? Again you have to filter it. Fine. So this hot saturated solution you have to filter. So this filtrate you will get. Now this filtrate you have to just kept in open without disturbing it for 1 or 2 days. And what you will see, children, that when it gets cool at normal temperature, it starts crystallizing. You will see crystals of the compound which are the purest form, and if still some impurity is left, it will remain in the liquid form, but the solid pure part will get crystallized, it will convert into solid form. You can do it at home by using salt which you are eating. Okay, so mix that salt. Mix means more solute has to be added in the solvent, right? Then heat it for 10 to 20 minutes, and after that filter it, and then keep the filter rate in the open for 2 to 3 days. You will find good crystals of sodium chloride, NaCl, salt which you are eating. So this experiment you can do at home. So this is the method of crystallization in which an impure compound can be converted into crystal. Crystal is the purest form. Okay, and impurities cannot solidified impurities cannot convert into solids. Okay, so impurities will remain in the solution if some soluble are still left, and pure compound will be changed into crystal form. Okay, now let's move on to the next slide and see that crystallization is a better technique than evaporation. This question can come. Why we are saying that crystallization is a better technique than evaporation? It is so because in evaporation what we do, we just the solution is heated to dryness. Okay, so in evaporation we kept it on the gas or we kept it in the sunlight, and the solution is heated for a long time, due to which sometimes what happen, that solids can decompose. Okay, the solid molecules of the compound, they can decompose, the molecules can break. So this is an negative point of evaporation. And in both the techniques what we do, that the solution is filtered. Fine. So in evaporation some soluble impurities may remain along with solid. Okay, whereas in crystallization what have in evaporation what we did, we just evaporated, so water goes out, but still some impurities, soluble impurities, can remain with the compound. But in case of crystallization what happens, if some impurities are still remaining, so they will remain in the liquid form only, and only the pure compound will get solidified. So these are the crystals, you can see these blue color crystals. So this is by crystallization. Okay, so impurities remain in the dish below, and only the solid pure form get crystallized. Whereas in evaporation, these crystals can damage, impurities can still remain with it, because we are not forming crystals, we are just evaporating the liquid. Okay, so solvent will evaporate, but the impurities can remain with the solid form here. So crystallization is much better technique than evaporation. Okay, now next is purification of drinking water. This is the last topic of the lesson now. So after this the lesson is over. So how the drinking water is purified? So here is the reservoir where the which is the main source of water from the from where the water is coming. So water is allowed to come through the reservoir, and firstly the water comes in the sedimentation tank, right? So in the sedimentation tank what is done, the water is allowed to settle down, right? Means water is allowed to stand without disturbing it. Then what happen, if any sediments, means sand particles which are heavy in nature, so they will all settle down here, they will all settle down, and the solid particles, heavy solid particles, will settle down at the bottom, and this is known as sedimentation. You have studied these techniques in 7th standard, right? So the heavy particles will settle down, that is called sedimentation. Okay, so this happens in the sedimentation tank, that all the heavy particles will settle down. Then water is allowed to move on to the loading tank. In the loading tank, alum is added to water. Alum, which is also called fitcurry, right? So this alum, what it will do, that all the particles, fine clay particles, they will get attached to the alum. Okay, and will sit along with the alum only. So here the suspended impurities, suspended means which were freely floating in the water, they get attached to alum, and in this way the water gets little more purified. After this loading tank, the water is allowed to move in the filtration tank, right? So now in the filtration tank you can see three layers here. Okay, so the lowest layer is of coarse gravel, then above it there is a layer of fine gravel, and then above there is a layer of fine sand. So water is allowed to come in this chamber. Okay, so in this chamber what happens, water is purified, water is filtered in this tank. When it passes through these three layers, all the impurities remain in these layers, and water get completely purified, from where the water is then allowed to go in the chlorination tank, right? So in this chlorination tank bleaching powder is added. Okay, and bleaching powder consists of chlorine, so this chlorine will kill the bacteria. And then afterwards, from this chlorination tank the water is supplied to all the houses. Okay, so this is the complete process of purification of drinking water. Fine. So firstly, the water is allowed to go to the sedimentation tank where all the sediment settle down. Then water goes to loading tank where alum is added. So the fine particles, suspended particles get attached to it. Then thirdly, the water is allowed to move in the filtration tank where the water is filtered with the help of three layers. And after that, the water is allowed to go in the chlorination tank where bleaching powder is added, and finally the water is then supplied to the homes. So this is all about the purification of drinking water. Now let's see what we have done today. We have seen sublimation, where the sublimatory substances directly change from solid form to gaseous form. Okay. Then chromatography, in which the colored components can be separated using paper, chromatographic filter paper, right? Then distillation, in which two miscible liquids which have a difference of 25 to 30 to 50 degrees Celsius can be separated. Then fractional distillation, in which the miscible liquids having very close boiling points can also be separated. Then crystallization, in which the impure compounds can be changed into crystal forms. And then the purification of the drinking water. So here the chapter is complete, children. If you have any doubts in any topic, in any subtopic of the lesson or the separation techniques we have done, you can ask anytime. Please feel free to ask the queries. Okay, if you'll not ask the queries, your concepts will not get clear. Okay, so we'll meet in the next session. Till then, everybody do watch the videos completely and try to understand the concepts. Okay, okay, stay home and stay safe, everyone.